hello, everybody. Welcome back. Last scientist to roll in. Hello. So welcome back. Happy to see so many of you coming back despite the beautiful weather outside. And I also want to welcome Dr. Darek Gorecki, who's here today. And he's going to talk about gene therapy. So as we mentioned earlier, our motivation is to always also inform about general, where is the field in terms of gene therapy. And we touched on that earlier, that also, you know, where is the field in terms of stem cells. And so I'm very happy that we, Darek has agreed to give this lecture. So a little bit about his background. So he's an MD, PhD, and is a professor of molecular medicine at the University of Portsmouth. And his main interest lies in Duchenne muscular dystrophy, which is also a rare disease and incurable, and it's a neuromuscular disease by the lack of the protein called dystrophin. So his team discovered that there's a receptor, which is a really good therapeutic target in Duchenne muscular dystrophy. He also described for the first time the distribution of different dystrophins in the brain, and also discovered the cerebellopoukinia non-specific dystrophin. So he's a specialist with knowledge in gene therapy that he's going to share with us, and he authored over 100 research papers. And yeah, so thank you very much for agreeing to come here, and we're looking forward to your presentation.
Thank you very much. Ladies and gentlemen, thank you very much for inviting me. It's really a great honor to be here and to be able to talk to you about, well, gene therapy. It would be more an introduction to gene therapy. I will try to dwell on some of the important aspects when thinking of developing a treatment based on genetic abnormality. There's this story about Albert Einstein teaching at Oxford. It's almost certainly untrue, but it illustrates my point quite well. It is Albert Einstein being chased by his assistant, Dr. Einstein, but isn't it the same exam question that you like the same group, that you asked the same group of students last year? Yes, it is. But how come? You know, how can you do that? Well, you see, this is the same question, but the answer has changed. And that's how science very much works. So it's still the same question. Is gene therapy able to revolutionize treatment of human diseases? And if I had this talk 2 years ago, I would probably do a lot of hand waving, and I would answer yes, probably, very likely, but we still need a lot more to do to have this answer. Today, I can say that almost certainly has already revolutionized the treatment of human diseases. But before I go into gene therapy, I would like to start with explaining what kind of approaches we can take to treat the deficit, the abnormality that occurs inside a cell. So obviously, cell is a very complex structure with all those intracellular organelles. And the most commonly used approach that we are all very familiar with is pharmacotherapy. So if there is a particular receptor or channel that we can design a small drug, small molecule that will bind this receptor or channel, the protein on the cell surface, and this way we will, after administration, achieve some therapeutic effect. The second approach is to replace the missing cell or defunct cell with a healthy copy. So bone marrow transplantation is nothing more than cell therapy, okay? Yet another way is a protein replacement. So if there is a protein missing, then the easiest way, one of the easiest way of doing the treatment would be to replace this missing protein. And of course, treatment of diabetes, administration of insulin is the best example of that sort of treatment. And finally, the most advanced, or perhaps the most complicated, but at the same time the most permanent treatment would be gene therapy. Would be either replacement or repair of the missing gene that is responsible for the abnormality that we observe inside the cell. All right. So what is gene therapy? There are two definitions that I put here quite deliberately, and I will read them out. So it's a procedure aimed at replacing, manipulating, or supplementing non-functional or misfunctioning genes with healthy variants of these genes. But it goes further, because it could be a treatment, but it also could be a prevention, in the case of vaccines, I will talk about it in a moment, of diseases by the transfer of therapeutic genetic material. It could be either desoxyribonucleic acid or ribonucleic acid, into a group of cells. Here we are talking about a tissue or an organ, to correct or modify, and could be up or down regulate the gene expression that is influencing the disease process. Okay, so historically, gene therapy started quite a few years ago. It was in 1980 when the first treatment has been tried. It was unsuccessful and it has been completely disregarded. Okay, because no safety protocols were obeyed in this treatment. The first successful gene therapy took place in 1991. Dr. Blees treated this little girl, who now is an adult woman, Ashanti da Silva. She was suffering from severe combined immunodeficiency. It's a disease in which the immune system does not work, so it's lethal. Any infection can kill such a child. And she got a specific gene therapy treatment in which her bone marrow stem cells were treated. So she is, I believe, still alive and well now. Now, since then, we went through many ups and downs, right? Around the 2000, we have, year 2000, we have a quite devastating a clinical trial in which young boy, Jesse Gelsinger, died. And since then, many of the clinical trials has been terminated. Centers were closed. It really seemed like gene therapy is going to be dead and buried. But then it started picking up, and we went through bust and boom. So one of the examples, the boom, is that the same gene therapy treatment for severe combined immune deficiency that was first tried by Blees on Ashanti was then picked up by the big pharma. And if big pharma, the multinational drug companies, are taking on something, it means that probably they sense that there will be some big money in it, so it's going to be successful. But at the same time, we had examples of a real bust. So the first gene therapy clinically used both in the US, worldwide, but also in the UK, first approved for clinical use, then was discontinued because it turned out that the clinical effects were not sufficiently good. There also the same happened to a treatment for the disease that I work on, Duchenne muscular dystrophy, that it was very promising initial outcome of the trial, and then it did not mean its outcomes, so it was discontinued. And then, of course, we had a very big boom, which was the COVID vaccines. So all of us, except those who did not get vaccinated, are subjects of gene therapy treatment. I'm not sure you realize that, okay? Because both the AstraZeneca and the Novartis vaccines were really a gene therapy products. I will explain how it worked in a moment, okay? Quite recently, I'm talking about a couple of weeks ago, there was an announcement that redosable gene therapy has been approved, okay? This is for this particular epidermolysis bullosa, this particular disease. It's a skin treatment. But what is important, you will understand why dosable gene therapy is such a landmark, hopefully when I get to the end of my talk, when I explain the problems with gene therapy. Okay. So before I start talking about gene therapy, I have to really get you to the beginning of the road where it all started. So we have a disease with genetic component, just like PURA or DMD. All right. So of course, in the past, it could take very long time for Huntington gene. It took up to 10 years to identify the gene that might be responsible for the disease. Then once the gene was identified and cloned, only then is possible to understand, to try to understand the biological defect that is caused by this disease. And again, as you've heard, PURA, its significance, its molecular mechanism is still not fully understood. So that stage can take quite a while. And of course, once we understand the biological defect, it might be possible to understand the downstream abnormality, which can perhaps be treated with drug therapy, right? Another possibility, of course, is to use the identified gene for gene therapy, for gene targeting. And finally, once we understand we have the gene, we could develop prognostic and diagnostic tests, how do the surveillance or prenatal diagnosis in some cases. So this is where we start. And of course, we are in a place that's revolutionized the identification and isolation of genes, okay? Because it was the human genome sequencing that changed it all. Now we have a sequence of a human genome, which means that instead of spending 10 years identifying the gene that is responsible for the abnormality, we can just request it and it will be sent to us in an envelope, in a little tube. So the things that we found were quite surprising from the human genome sequencing project was that we are not having any more genes than many other organisms, a mouse or a fish. The complexity of human beings is not in the number of genes, but in the gene regulation. And of course, PURA protein is an example of a protein that is responsible for this complexity because of its involvement in regulation. Also, you will notice that we still don't know functions of about 50% of the genes that we have actually sequenced. So I know that you had some introduction into a transcription and translation, how the gene works, what's the difference between the protein and genetic material. But I would like to go back to it and perhaps introduce it in a slightly different way. So if we say that our genome is a blueprint for life, of course, every cell has the same amount and the same number of copies of the genetic material. So in each cell, the blueprint is the same. And it resides in the desoxyribonucleic acid inside the part of the cell. So if this is the cell, it resides inside the part which is called nucleus. All right. And it means that every cell has the same blueprint, but each cell may need only a subset of genes, only some of the information from this blueprint to function normally. So then from this blueprint, we have to get some sort of a instruction manual, so how to assemble a specific cell. So that happens in the process called transcription, where this genetic material, as a genetic information and DNA, is transcribed into this manual, instruction manual in a form of another type of nucleic acid called RNA. And this is specifically messenger RNA. It carries the message how to do things. All this happens inside the nucleus. That's what I need to stress very strongly. And then messenger RNA is exported. There are natural mechanisms that allow the messenger RNA to go out into cytoplasm or some specific organelles inside the cell, and there it is translated into the building blocks of a cell, which are proteins. Very few proteins are coming back into the nucleus, and this mechanism of translocation to the nucleus is very important. I show this because I understand that PURA is functioning both in the cytoplasm, or mostly in the cytoplasm, but some of it needs to get into the nucleus. And it's quite interesting to understand what is the mechanism of this translocation for PURA to the nucleus. Now, this is what happens when everything is all right. And then a mutation happens. Okay, so we have a large number, about 10,000 diseases which are caused by single gene mutations. And of course, there is a growing number of polygenic traits when multiple abnormalities in multiple genes works together. So that is where we are thinking of what could be done at the genetic level when we have a mutation, when this normal blueprint is no longer functioning as it should. Okay, and here comes the gene therapy. So again, it aims to treat human diseases by transfer of genetic material into specific cells. That's important. But we have to remember that that's a very modular technology. So the outcome of gene therapy can vary greatly. So obvious thing is a correction of the genetic defect, but gene therapy can be used to introduce a new function in a cell or to activate genes that are perhaps active during the development, then switched off, and then a slightly better version, more up to the job version of a gene is activated. And then if this is mutated, then by reactivating this version that is active during the development, we actually could cure the patient, or at least treat the symptoms. And in a particular situation, we can even trigger cell death. And of course, you can guess rightly where we want to use gene therapy, where we want to trigger cell death. It's in treating cancer cells, right? So it is a modular technology. Another thing you need to appreciate, to understand, is that there are specific requirements for the development of gene therapy. So first of all, we have to know and we have to clone the gene. We have to have the specific genetic material available. Then we have to be able to target our therapeutic gene into the right cell, right? Or right tissue, or a number of cells. So first of all, we have to understand which tissue, which cell is affected by the abnormality. And then we have to be able to access this cell with our therapeutic product. So again, in case of PURA disease with syndrome, we know that the abnormality occurs in the CNS, in the brain, perhaps also in muscle, but clearly from the complexity of the syndrome, the disease affects multiple tissues. Right? Which means that it's a little bit more difficult to treat this kind of disorder compared to a disease in which just one type of cell is affected. And another thing is that we have to be able to control the level of expression of the affected gene. The simplest example is diabetes. We could do gene therapy for diabetes. Instead of injections, the patient could be given genetic treatment. But the problem is that it could be very dangerous because too little insulin is bad. It causes diabetes, but too much insulin would kill the patient. So getting the right level of expression is absolutely vital. Again, PURA protein, we have to be very careful not to create a situation when too much is much worse than too little. We have to understand the level of expression. Now, how do we do that? Because, you know, I'm sure you've heard that we take the therapeutic gene and we put it back into a patient and, you know, that is gene therapy. But it starts with actually the bacterial DNA, right, which is called plasmid DNA. So inside each bacterial cells, there is this little circular DNA, which bacteria use as a communication tool. They swap plasmids, and for example, swapping those plasmids that encode antibiotic resistance, they acquire antibiotic resistance through different bacterial strains. So those plasmids can be easily isolated, and then we use this as a backbone for the creation of a therapeutic construct. So how do we do that? We take the plasmid DNA and we modify it in such a way that instead of the bacterial genes, we put our therapeutic gene, our transgene. All right? I will explain in a moment how it's done. We have two more elements to be added to the therapeutic construct. They are called promoter and enhancer. Promoter, to put it simply, is the switch on and off, which allows this therapeutic gene to be activated. An enhancer is the tuning of the volume adjustment, right? So using specific combination of promoters and enhancers, we can express the therapeutic gene only in a specific cell in which this particular promoter is active, and then we can adjust the volume of expression, level of expression, by using specific enhancer, right? And that combination, making this construct, is possible thanks to the work of these three gentlemen that got a Nobel Prize for this discovery of recombinant DNA. They discovered that there are enzymes that are able to recognize the specific sequence. The DNA have 4 letters, right? And the combination of those 4 letters are recognized by number. There's hundreds of different enzymes that recognize just a specific sequence. They can cleave it. Then we mix the human genetic material with bacterial genetic material. We mix them together. We not add another enzyme, and we make recombinant DNA. In this case, this bacterial plasmids contains all the elements that I explained before: the transgene, the therapeutic gene, let's say PURA, with the specific controlling elements that are added into it. And then we can transfect bacteria and produce large amounts of this plasmid DNA. Additional technological breakthrough was the polymerase chain reaction, which again is Nobel Prize in 1993. Of course, you've heard about PCR because all the COVID tests were done using PCR. So I'm sure that you appreciate that polymerase chain reaction means that we can start with 1 copy, and using specific enzyme, we can produce multiple copies of the let's say therapeutic gene that we want to have. So it makes our life a lot easier. And of course, you would like to believe that at this stage, we just take this purified plasmid DNA that contains this therapeutic construct, and we just inject it, and that's what gene therapy is about. But I'm afraid that's not what's going to happen, because the cell or our bodies have evolved over the millions of years of evolution not to take up foreign DNA, whether it's a plasmid DNA or any other form of DNA. Why? Because, of course, this kind of DNA usually means that it's infectious, right? It's either viral or bacterial. So there are specific enzymes that would break down this kind of DNA, therapeutic DNA that we inject. None of it would have a chance to get into the cell. And even if it gets in contact with the cell, it still would not be able to go in. It's a very big molecule, and it has to be modified, all right? And this modification is required to allow its entry into the cell. Again, it could go into the cell, but it doesn't mean that it would be able to do its job, because this kind of molecule that's taken by this process called endocytosis is degraded, right? Because the cell needs the building block. It doesn't need those molecules here. And what's not degraded is kicked out of the cell. So very few molecules actually have a chance to get out of those vesicles here. And even then, they would not be able to do their job because this plasmid DNA encoding the therapeutic with the therapeutic gene needs to get into the nucleus, navigates its way into the vicinity of a cell nucleus, because as you remember, I told you that all the transcriptional machinery is sequestered, is kept inside the nucleus. So the difficulty is to get this therapeutic gene into the nucleus when it can be transcribed and then translated into a therapeutic protein, PURA. All right. So, of course, we need some sort of a Trojan horse. We need a way to get the therapeutic gene into our cells. And there are the systems that are used to transport nucleic acids into our cells are known as vectors. There are two types of vectors, the viral and non-viral synthetic types. Now, there is a number of viruses that are absolutely fantastic at getting their nucleic acid into our cells. They not only are efficient, they evolved through evolution to do it, but they also can do it in a very specific way. So let's say adenovirus can infect respiratory cells. Lentivirus can affect blood cells, can infect blood cells. Herpes virus would be able to affect nerve cells. So by using specific virus tropism, we could target our therapeutic gene to specific cells. And of course, there are also non-viral synthetic vectors. Ideally, we would like to have a vector which has properties of the viral ones, but are entirely synthetic. Okay, so that is the work under development. And the important thing is that not all viruses can be used because their cloning capacity to take the therapeutic gene size-wise is limited. But I would like to make something sure. When we are talking about viral vectors, we are not talking about viruses. Okay, so virus will contain the genes that are needed for the virus to produce more viruses, to replicate, all right? In a viral vector, all these viral genes that are allowing viruses to propagate and produce more variants are removed and replaced with the therapeutic gene. Okay, so we just have a shell that allows the targeting, but we don't have any chance of infection resulting from using the viral vector. So please remember that that's a very big difference. All right. And then another part of the toolbox is to explain how can we do those modifications. So there are two ways of doing it. Of course, ideally, we would like to take our therapeutic construct packaged into a viral or non-viral vector, inject it into a patient. It would find its way to a specific tissue in which the expression is needed, and we will have a therapeutic effect. Another way of doing it is to isolate the cells that are affected by the genetic disease, culture these cells outside of the body, modify them using viral, non-viral vectors and other methods, then selecting those cells that have been modified and are in a sense healthy, and then putting them back into the body, into the tissue from which they came out. All right. Now, we don't have our own stem cells available when we are adults. So it's not surprising that these two gentlemen got Nobel Prize in 2012 for developing cells that are taken from our own adult bodies, but can be turned into stem cells like those that are shown here, let's say, taken from the bone marrow. All right. So these induced pluripotent stem cells are yet another way of doing the targeting of a therapeutic gene into specific tissue, into specific cell. Another thing I have to stress is that there are certain things we can do and there are certain that we cannot do or should not do. So any modifications that that could result in a genetic modification, transfer of a genetic material to get into germ-line cells are not allowed, all right? Because this means that such modifications would be passed to the next generation, and we don't want it. We don't want to tweak with human genetics, human genome to that extent. What we can do is so-called somatic gene therapy. In this case, the cells that are modified could produce the therapeutic effect, but this effect will be terminated with the end of life of a patient that we treated. Okay? But there is an interesting question about at which stage we actually could do the gene therapy. So as I said, germline gene therapy is not allowed, but how about gene therapy in utero, fetal gene therapy? Okay, would it make sense to start the genetic modifications when the baby is still in the womb? And of course, it is allowed, because as long as we do not modify the gonads, this modification will still be somatic cell therapy, which means gene therapy, which means that all the modifications will cease to exist when the baby grows up, becomes an old but still healthy individual, and then die of natural causes, right? You can imagine that if we are talking about skeletal malformations or inherited blindness or congenital diseases that affect cognition, then doing it early on would prevent the onset of symptoms rather than treat the symptoms when it might be too late. There is yet another reason to do it, because this could also become tolerogenic. I will explain in a second. This is all fine until we start really doing it in humans. Because first of all, the viruses are excellent at targeting their genetic material, even if they are modified, so the therapeutic gene, into our tissues. But they're also extremely immunogenic. We all come across those viruses that we use as vectors, which means that most of us, most of the subjects of gene therapy, patients, would have pre-existing immunity, which means that we have to modify those viral vectors to avoid immune system recognizing them, exactly the same way as our immune system is fighting infections, viral infections, right? Then, of course, some viral vectors could cause acute toxicity. And finally, over time, it is likely that cells that were transduced with the viral vector will be seen by the immune system and destroyed. So the persistence of the effect may be very much determined by the fact that there is immune response developing. Even pure DNA, as we have shown, can accelerate autoimmunity, all right? So there are diseases in which DNA is being recognized as foreign, and if we, in individuals who are predisposed to this kind of autoimmunity, use gene therapy product, we can actually trigger even more autoimmune responses. So that is one of the problems with using gene therapy. There is yet another problem. This is the, well, immune response or immunogenicity, as we call it, against the therapeutic gene product, all right? In some cases, it's brilliant if we want to use it for vaccination, but if we want to have long-term therapeutic expression, then I'm afraid it's a very bad news. What I mean by that is I've already mentioned that we all been subjected to gene therapy treatment because the COVID vaccination was exactly that. So what happened is, if I put together all that I told you so far and package it into those vaccines, is that AstraZeneca had a viral vector containing the cassette, the expression, the DNA that encodes the spike protein from the COVID virus, right? The Pfizer and Moderna, they use RNA. So you remember transcription, translation, it's still the coding nucleic acid, but it's a step down from the DNA, which means that the AstraZeneca virus needed to get its therapeutic construct into the nucleus, while the, let's say, Pfizer or Moderna just needed to get into the cytoplasm, where this messenger RNA was translated into the spike protein, and that triggered the immune response. So that's how the gene therapy, gene therapy was used to vaccinate the people against COVID-19. All right. Now, this is all wonderful news for vaccination, but if we want to have a long-term expression, then I'm afraid then in some situations where there is absolutely genetically void patient who does not express any protein, then of course this this patient, when injected with the vector that expresses this therapeutic protein, its immune system will see this therapeutic protein as foreign, non-self, which means that it will trigger the immune response exactly the same as COVID vaccines triggered response to the spike protein. And that's an example. This is just 2 days ago. There is an approval of a gene therapy for Duchenne muscular dystrophy, but it is limited to patients who have on specific mutations only. Because if they use it in some other patients who do not express any dystrophin, that it led to a tragic consequences. In 3 out of 4 clinical trials that were run last year, 3 ended up with a very severe side effects, with 2 patients unfortunately dying because the immune response against the therapeutic protein that was used in those trials. So as you can see, there is a number of problems that we have to solve. Now, last thing I want to talk to you about is what we need to achieve, what we need to do if we want to treat a disease that has genetic background. So in this case, we are talking about a normal situation. Each of us have 2 alleles, two genes coming from a father and a mother, and they are translated into proteins that can, let's say, work together as a group, and they are fully active. Now, in a situation when we have a recessive mutation, it means that both alleles must be mutated, both genes must be mutated. There is no protein made there, and therefore we have a disease. The most complicated situation is when we have a dominant mutation, which means that the mutant gene acquires a new function, and this function can dominate, can take over the normal protein that's still there, but is non-functional, okay? So in the case of PURA, we have haploinsufficiency, which means that one allele here should be sufficient to overcome the mutation in the other allele, in the other gene. But unfortunately, it is not. So therefore, adding extra copies of a normal gene may be sufficient, but if there is some dominant effect of the mutant PURA protein, then this effect may not be sufficient. So in this case, we need to use different tools that would allow destruction of the mutant messenger RNA. Remember this instruction for making specific proteins. And there are ways of doing it. One way is to using those morpholine oligos, short fragments of DNA that trigger, either block the translation of the mutant gene or trigger its degradation. And the most advanced is using CRISPR-Cas, where I'm not going to go into details. But again, 2 years ago, these two ladies got the Nobel Prize for this discovery, because really it's a game changer in genome editing. So it would allow us to repair the mutant gene in its site, in its locus. All right. So the question is gene therapy really a panacea, a treatment for all diseases, or in some cases it would be a placebo? We still have to improve the vectors and delivery methods. So ideally, we would like to use non-viral synthetic vectors to avoid immune responses. Perhaps stem cell gene therapy would be a way forward. We have to remember that in many cases, we will have to prevent or combat immune responses against the therapeutic gene, which becomes an antigen. And we have to be absolutely able to control the level of expression. Otherwise, it may do more harm than good. With that, I would like to end, and thank you very much for your attention.
Thank you, Dr. Gorecki. Is there any questions on the floor here? I do have some online, but. Okay, I will go to my online questions. What can we expect from gene therapy at PURA syndrome specifically?
Well, I think I have to manage the expectations. I've tried to explain that in the case of PURA, we do not have a full understanding of the underlying molecular deficit, that the problem will be also targeting all the tissues, all the cells that are affected by the deficiency, by the mutation of the PURA gene. And there may also be a question of the level of expression that would need to be very carefully titrated. So you see, the reason why I mentioned this anecdote is that what we know now and what we can say now that is hard or impossible, it may be resolved in a month or a year. So I would be very optimistic, because the progress that we've observed in gene therapy in the last year or two is absolutely tremendous. So
what is your opinion about vaccinating a baby with PURA syndrome with the MMR vaccine?
I have no comment on that. I have no reason to believe that it would be in any way detrimental, but I would not like to comment further than that.
Okay. We have stem cells from an older, healthy child. Could we use it for any good purpose for our younger child with the diagnosis of PURA syndrome?
Well, providing that the child has, it's genetically very similar. Otherwise, it would be like transplanting an organ from one sibling to another. So it could be seen as non-self and rejected. Exactly the way I explained why the therapeutic product can be seen as foreign.
Hi, probably a stupid question, I'm sure, but if you could achieve success with any disease with the gene therapy, what could that look like? What sort of difference could that make? So what sort of level of recovery could a child with PURA have, for example?
Well, any disease, you mean? Any disease. Okay. All right. As an example, in spinal muscular atrophy, the most severe form is so-called floppy baby. The baby is absolutely unable to breathe unaided, let alone move. With gene therapy that has been developed recently, these baby can sit, and some of them perhaps even walk. So that would be the level of expectancy that I would have for successful gene therapy.
maybe you comment on neuronal plasticity, because it's a little different than muscle, right?
Well, in this case, it was actually neuronal. It was spinal muscular atrophy is caused by the motor neuron. It's a motor neuron disease. So this was a treatment of motor neurons in the spinal cord, which then affected the muscle. Muscle needs to be stimulated by neurons, otherwise they cannot survive. So that was quite a spectacular success. Right. There are some diseases, obviously, that are easier. Hematological abnormalities are easier to treat with gene therapy because of the bone marrow stem cells that can be used, that can be targeted. Far more difficult to do it with brain abnormalities, in which case we not only will have to go into a specific, not into just going into the brain, but perhaps to hit the right neurons in the right part of the brain and then express the level and then express the protein at the right level for these neurons to function normally. Right. So that's this additional level of complexity.
Julie, do you want? I saw your.
As PURA obviously still new, even though my son's 23, he wasn't diagnosed until he was 18. And from the day that we left the hospital, they were saying there's chromosome 6, is a spelling mistake. So when I look at what you're saying there about babies, how putting that into them when they're younger as a fetal, at what point then do you actually know that someone's got PURA, especially when we didn't know until he was 18 and nobody knew?
I understand that now there is a test for PURA, but I'm not sure it's being used in a prenatal diagnosis. Well, I'm sure not in a general population, but I'm not sure if it's done even in the families.
No, so I mentioned this earlier on today. So in some instances where a couple is pregnant and there are concerns about the pregnancy and an invasive test of the pregnancy is performed, rather than just doing conventional chromosome tests and karyotypes, there are now in the UK at least rapid whole genome sequencing of the fetal genome, of the baby's genome. And so we are making diagnoses of conditions that in the past, molecular diagnoses that wouldn't have been made, because we're using this technology where we're sequencing DNA rather than just looking at chromosomes. But this is only in the context of pregnancies where concerns are raised that warrant an invasive test. And so for what are thought to be healthy pregnancies, and I think most parents of children with PURA syndrome won't have had significant concerns flagged during the pregnancy from combined tests or from antenatal imaging, and there would be no prompt to investigate that pregnancy. So at the moment, most cases would go undiagnosed until birth, when it is clear that a baby is born and they are floppy and have problems with breathing, possibly with apneas or with establishing feeds or having seizures in the neonatal period or infancy. Clearly, once a child is born, there is then the opportunity to do this testing if there weren't any concerns antenatally. And as was mentioned earlier, there will be in the UK a newborn whole genome screening program, which is going to be trialed at some centres, I think, from the end of the year or beginning of next year. But the plan is that this will be rolled out and that essentially all children born in the UK could have whole genome sequencing at birth if their parents agree to that. So it does mean that we probably will end up diagnosing lots of things earlier on in life. There are lots of ethical questions about this that we clearly don't have time to go into right now, but it's a big debate nationally. But in terms of identifying something sufficiently early that you could use a therapy antenatally to improve the outcome, well, clearly the sooner you can identify a problem, the sooner you can deliver an effective treatment, the better the outcome would be. But we don't have a therapy that we could offer at the moment, so it is all theoretical. But if there were a good gene therapy, then yes, you would want to make that diagnosis as early on as possible, and potentially treat in the womb for the best results. That would be the theory, because it is a neurodevelopmental disorder as we understand it. So if the brain starts to develop in an abnormal way, even if you have a therapy that you can administer at birth or in infancy, it may be that some of the early development that was abnormal cannot be corrected. So, if we're thinking about gene therapy, then treatment earlier, particularly for neurodevelopmental disorders, would be preferable. So again, a very long-winded answer, really sorry, but hopefully it makes sense that.
But then with that whole James and my sister, so she be entitled to have, when she gets pregnant, have some sort of test that would affect baby. I know it's not ready to have the same who actually then gets the chance to take a look at being checked out in that way.
So at the moment, there wouldn't be testing antenatally unless there were concerns about the pregnancy. But no, exactly. Yeah. So this is.
Then a sibling has a brother who has got that already.
Yeah.
Would be able to say that I have concerns with my brother who has got it. Would you then be able to give me?
Not at the moment, no, because we would say that the risk of having a child with PURA syndrome would be at the general population level, because she doesn't have it, so she can't pass on that genetic change. So if it were to happen, it would be by chance alone, and the chance of that happening would be very, very small indeed. And if she were to have a child with PURA syndrome, it almost certainly wouldn't be a child with exactly the same genetic change, because she doesn't carry it. So that would be a really unlucky coincidence if that were to happen. But in practice, it's so unfathomably improbable that it isn't really worth being terribly concerned about, because there are other things which are more likely in a pregnancy. So despite the family history, things like chromosomal abnormalities would be a greater risk than something like PURA syndrome in offspring. But you know, it is a landscape that's changing very quickly, because technology is moving on so very fast. It's conceivable that in the not too distant future, it will be possible to offer fetal genome testing on maternal blood. At the moment, although it exists as a proof of principle, it isn't something that is accessible. But presumably there will be an argument for that approach if we're thinking particularly about intervening antenatally and offering treatments potentially in the womb that could change the course of disease for someone. But of course, those therapies have to exist.
So I also have a question. So I think you pointed out already that, you know, if you want to affect every cell in the body, that's of course a challenge. But for example, the brain obviously is difficult to reach, but if one would want to target the muscle, how do I have to envision that? Would you get intramuscular injections of the virus and then just have your right biceps being targeted, and then you go on the left biceps? Or and then what is the reach of that one particle injection then?
This is the question that we wait and that we have to face when we think about gene therapy for, let's say, Duchenne muscular dystrophy, in which every muscle except extraocular is affected by the disease. And of course, doing injection into each muscle is not possible. And even injecting each muscle would not be enough, would not give enough coverage of the muscle cells. So what is being tested is the use of a vector called adeno-associated virus, AAV, that has a very, very strong tropism is very good at recognizing and getting into its genetic material into muscle cells. And that's being done by intravenous injection with the brief disruption of the blood vessels, opening of blood vessels. The virus injected intravenously can get out of the bloodstream and gets into muscle and then targets a majority. So it's a very effective way, and that's how this gene therapy is being done.
Yeah, I wanted to start by saying that it was great to see, to hear this very balanced view on the pros and cons that these technologies bring along, because I think this is one of the main issues we're discussing over and over again. So that was really good and helpful.
Thank you.
The question I have is more a scientific question again, like Tina's question, but perhaps turning around a bit. So you mentioned already the difficulties of having the right expression level, the difficulties of immunogenicity, and then there's the problem to hit all the cells in the body. If we would make that a little bit smaller and say, oh, well, it's primarily a neurological disorder, at least from what you know from now. By now, there are some vectors, viral vectors like AAV serotype 9 that are neurotrophic, meaning they like to enter neural cells. Could you give us a status report on where we are with this? Is this, I think there are some clinical trials with that, at least animal models are available for that.
In animal models, there have been some quite tremendous progress using various viral vectors, and AAV serotypes, specific serotypes is one example. Herpes virus is another one that is more peripheral nervous system. But nevertheless, I don't know of any, I don't remember of any clinical trials that have been initiated for this, unless they are very small scale and a pilot studies. So we're not there yet. I think that's quite important to remember. I mean, please remember that the first thing we have to bear in mind is that we should not harm the patients, right? Primum non nocere. So we cannot try something that potentially may be good, but if we are not careful, it may cause more damage. And I think that's the reason why the CNS treatment is not that advanced. On the other hand, I just gave an example of SMA, which is treated by intrathecal, so injection into a spinal cord where it's distributed with this fluid up to the specific neurons, which motor neurons that are responsible for controlling muscle function. So it is not impossible, all right?
Anyone else? Okay. We're all set. Thank you, Dr. Gracke.